Hello, and welcome back to Opinionated Science. This is the podcast that's brought to you by Technology Networks. And if you've not joined me here before, my name's Lucy. I'm the scientific content producer here at Technology Networks. And for this episode, I will be your host. Today, you've joined me for a great episode because we're taking a deep dive into the world of culturing cells. So if we take it all the way back to basics, cell culturing is the growth of microorganisms such as bacteria and yeast or human, plant or animal cells in the lab. And these cell cultures can be used to diagnose infections, to test new drugs, and are also incredible tools for research. The first cell culture techniques were developed some 100 years ago, so you can imagine that since then they've contributed to some tremendous breakthroughs in science. But today it's just seen as a fundamental tool used in labs around the world, and today's guest uses them almost every day, Dr. Daris Widera. He is currently an assistant professor and lecturer at the University of Reading in the United Kingdom, and his lab is mainly interested in neural crest-developed stem cell populations, which are promising candidates for the development of autologous stem cell-based therapies. He further works on influence of injury and inflammation on stem cells and somatic cells, and develops novel approaches for clinical-grade 3D cultivation of human stem cells. I'll be speaking with Darius to explore how scientists can use cell culturing within their labs, the differences between 2D and 3D cell cultures, as well as looking to the future to see where this space is heading next. And just so you know, this episode is very kindly sponsored by Acrobiosystems. They are a leading manufacturer of recombinant proteins and other critical reagents to support the development of target therapeutics, vaccines, and diagnostics. The company employs an application-oriented development strategy with a particular focus on product design, quality control, and solution-based support. Acrobiosystems developed a series of highly active recombinant cytokines named ActiveMax, which are based on the HEK293 cells expression technology platform for immune cells, stem cells, and other cell cultures. ActiveMax cytokines includes Activin A, CCL19, erythropoietin, FL3 ligand, GCSF, GM-CSF, IFN gamma, IL4, IL6, IL7, LIF, oncostatin M, SCF, TGF beta, TNF alpha, VEGF110, VEGF120, VEGF121, VEGF164, and VEGF165. So get ready because you're about to hear my interview with Darius in full. Darius, first and foremost, welcome and thank you for being here with me today on the podcast.
Thank you very much, and thank you very much for inviting me. My name is Darius Widera, and I'm Associate Professor in Stem Cell Biology and Regenerative Medicine, currently working at the Reading School of Pharmacy at the University of Reading.
Perfect.
In addition to my duties as Associate Professor, I'm also Research Division Lead for the School of Pharmacy, and I'm sitting on Scientific Advisory Board for a biomedical company in Germany.
Wonderful. I actually had come across your lab when I saw an article on how you'd hosted a presentation at the Global Cell Culture Symposium, and you'd spoken about 3D culture of stem cells, and I'd basically just seen how well your talk was received, and it immediately became clear that you were someone I knew we had to interview for this podcast all about cell culture. So you've already touched on it slightly, but I just have so many questions for you. The first question I have is, I was wondering if you could explain a little bit more about your background and your research background in general.
Of course, more than happy to do it. My research background is actually quite diverse. My first degree is in biological sciences. Then I moved on and did my master's in biochemistry, my PhD in neurobiochemistry, and then finally ended up in stem cell biology in the research lab, which is actually the area which we are doing a lot of cell culture. We are relying on expanding cells outside of the body. In terms of my research, we are heavily interested in adult and embryonic stem cells, but in addition to this, we are also doing some routine cell culture, and we are working on cancer stem cells as well.
Wonderful. And I just wanted to ask you a further question, just touching on, I've been on your lab's website, but I just wanted to know what got you into science. Why did you think this is where I want to be, this is what I would like to be doing?
Well, to answer this question honestly, biology was always my strongest subject, and I always wanted to understand how basically the body works, how all of those organs in the body are connected, how those cells actually talk to each other, and how such complicated system is able to perform its daily duties day by day. And this was basically what motivated me to move on. I studied biological sciences, as I mentioned before, and during my studies, I was also working in the research and development laboratory of the German biotech company. And this is where I experienced cell culture for the very first time. And this was something I was very passionate about. And I think this is state of the art and cutting edge methodology, which can lead to development of new potential therapeutics for disorders and conditions that we don't currently have any cure for.
Lovely. And then that kind of brings us nicely to the fact that you are, as you said, an associate professor in stem cell biology and regen med at the University of Reading. You do a lot of work into cell culturing, but what actually is cell culturing? And I think it would also be really beneficial if you could tell us some of the most common applications for it. So, why do we do it?
Sure, more than happy to do that. In simple terms, cell culture is a method where we can keep cells alive outside of the body. And this can be any type of cell you can imagine. You can culture human cells, you can culture animal cells. In fact, you can also cultivate plant cells or even insect cells. And this is definition of cell culture. Under proper conditions outside of the body, we can keep cells alive and happy. They can divide and do what they would do inside of the body as well. They can fulfill specific functions. For example, we can cultivate nerve cells. We can study how those nerve cells communicate with each other. And if you think about translation and medicine, we can also try to understand why neurons or nerve cells stop functioning properly in disorders and conditions like Alzheimer's or Parkinson's. Cell culture, however, can not only be used to develop new treatment options for different disorders and conditions, we can use cell culture for production of biological medicines, for example, many vaccines. And this is something quite interesting given the ongoing pandemic. Many vaccines are indeed developed using cell culture technology. In addition to this, cell culture is often used for fundamental biology. If we want to understand how cells fulfill their function, we need to study them in isolation, since human body is intrinsically very complex. So it's very hard to understand how those different systems work in combination with each other, but we can understand very well how isolated cells work in isolation. In addition to this, we can also use cell culture to investigate toxicity and study novel drug compounds. And this is something incredibly important if you think about preserving animal lives. We can save a lot of animals because we can test many things already in a dish without the need of animal experimentation.
Fantastic. I really, really like that idea that actually we're not going to be needing to use animals when we can just do it in a dish. I think that will appeal to quite a lot of people. And can you talk to the three different types of cells that are cultured in a lab? Because they can be classified into three different types. So we've got primary cells, we've got transformed cells, and then self-renewing cells. Can you tell us more about these and perhaps the differences?
Of course, I'm more than happy to do so. As you mentioned before, there are three general types of cells that you could cultivate in a dish. And the first type, and this is quite common, are so-called primary cells. Primary cells are cells that you, for example, would isolate from the patient. You would put them into a dish and study how the cells behave. So those cells are primary cells. Key thing to remember here is that those cells, as the patient himself, are not immortal. They age even in a dish, so you can only study them for a limited amount of time.
Mhm.
This is incredibly useful because we can then think about personalized treatment options for certain conditions. However, the limitation is that after a while, those cells will basically die in a dish. Next type of cells that is commonly used in research, but also in development of therapeutics, are so-called transformed or immortal cell lines, and cells, and we refer to them as cell lines because they can be propagated for forever. They are not affected by cellular aging. They don't age. And some of commonly used cell lines in the lab have been actually isolated in the 50s of the last century. To be more specific, the most common cell line that we are using in the lab, so-called HeLa, is an immortal cell line that has been first isolated in 1951. So we have them in a dish since over 70 years, and they are still incredibly useful. We have nowadays plenty of different types of those cell lines, and they are usually isolated from cancer cells, and they are intrinsically immortal. What can also be done quite easily in the lab, we could take primary cells and engineer them to behave like immortal cells. We can immortalize them, and this makes them into a new cell lines, and we can generate cell lines from any organ that you can imagine. We do have neuronal cell lines. Those are cell lines that have been isolated from brain cells. We have cell lines that have been isolated from muscle, from liver cells, and all of those cells are incredibly useful if you think about toxicity testing, but also for understanding of diseases and also developing new treatment options. Lastly, I'm glad that you mentioned this, since you've been referring indirectly to stem cells. You've been talking about self-renewing stem cells. Those self-renewing cells are slightly different to primary cells and also compared to cancer cells. They are not affected by cellular aging, and this is especially true if you think about cells such as embryonic stem cells or induced pluripotent stem cells. They do not age, and they can specialize or differentiate into many different cell types in the body. If we apply the correct combination of chemicals and also appropriate conditions in the dish, we can force the cells to become something new. We can force a stem cell to become a neuron. We can force the stem cell to become a bone cell, a muscle cell, and so on. And this can be used within personalized therapy, hopefully in a decade or two.
And then, to be realistic, even an expert like you with an incredible group of PhD students, postdoc research associates, master's students, all within your team, of course you'll come across some trickier aspects of working with cell cultures. What have you found are the trickiest challenges when you're culturing cells?
Given the very different nature of the three cell types we have discussed before, the challenges and potential roadblocks for work with cell culture are very different. If you think about primary cells, cells isolated from the patient, then we have the problem of cellular aging. Again, we have only very limited time frame to work with those cells. After this time, the cells will die. So you need to be quick. Also, those cells are usually isolated from tiny samples from the patient, so we have only limited number of those cells. This is quite typical problem if it comes to working with primary cells. There is another challenge if you are working with self-renewing cells or stem cells. Those cells divide very quickly in a dish, and this actually carries an intrinsic danger. If cells are dividing quickly, and this is true not just for cells outside of the body, this is true for the cell in a physiological environment as well. If they divide quickly, the likelihood of developing mutations is increasing. Therefore, you need very established and tightly controlled quality measurements in the lab. You need to ensure that the cells you are working this week are the same as the cells you have been working 3 weeks ago. This is even more important if you think about those immortal cells that we often have in the lab for over 50 years. All of those cells evolve, they change in the lab, therefore quality control is extremely important. Also, Since in contrast to our body, our flask in cell culture don't have an immune system. We are always facing challenges in keeping our cells clean, keeping them sterile. And this is indeed a very common problem in cell culture, contaminations. Those human cells or animal cells are often contaminated by bacteria, they are contaminated by yeast, and also, and this is the most tricky type of contamination, they are often contaminated by mycoplasma. Mycoplasma are very, very tiny organisms. They are even smaller than bacteria, and it's incredibly hard to see them even under the microscope. So this is also quite important part of the quality control. You need to ensure that your cells that you are growing in the dish are pure population and not contaminated by bacteria, mycoplasma, or yeast cells that you potentially don't see. Lastly, if it comes to industrial application of cell culture, scaling up is actually a problem. This is something the industry is facing from time to time. It's very different to establish a small-scale cell culture protocol compared to the cultivation of cells in hectoliters. And to be realistic, those are the amounts of cells that we need, for example, if we produce therapeutic antibodies in the cell culture. We need hectoliters of cells, and development of those protocols is very challenging since the protocols and the techniques are very different if we compare large scale and small scale in a research lab.
So something I've seen quite a lot of recently, and I'm sure our listeners will also have had some awareness of this, is the fact that you can have 3D cell cultures and 2D cell cultures. What are these and how do they differ?
This is an extremely good question, and I totally forgot to mention this. Traditionally, if we culture our cells in a dish, we cultivate them on flat plastic or glass surfaces. Obviously, if you think about a human or animal body, those are not flat. However, if we cultivate our cells in 2D, we are forcing this unnatural polarity. We are putting them on a flat surface. And this changes actually quite a lot. This changes fundamental cellular properties like the speed of division, also what the cells can do in terms of differentiation. All of those aspects are changed. And the reason is that cells in the body are regulated not just by biochemical signals, they are also regulated by physical cues, and this is something what is very often underestimated. Those physical cues include contact with other cells, contact with so-called extracellular matrix, those are proteins that are on the surface on the cells, but also by stiffness and micro and macro topology of the surroundings. If you think about organs such as a bone, obviously, this is a very stiff and rigid surrounding compared, for example, to a muscle or fat tissue, which is soft. All of those cues dictate how the cell is behaving. If we now remove the cell from the body and put it on a glass or plastic surface, obviously those fundamental cues are removed as well. This leads in change behavior of the cell. This can be at least partly avoided if cells are embedded in hydrogels. Those are very soft substrates where you can cultivate your cells in. So you sort of mimic the physiological 3D surroundings of the cell. And this has many advantages. Again, in terms of biological properties, we are more closer to the living organism. We can also preserve the polarity of the cells and avoid all of those common problems of cell cultures like changed morphology, changed cell behavior, and so on. However, there are also drawbacks. 3D cell culture, in simple terms, is not very cheap. Also, the training required for staff handling those 3D cell cultures is more intense. And lastly, the automation is less easy than if we think about 2D cell culture. Overall, 3D cell culture is something very timely, and many not just research labs, but also industrial labs are now actively exploring the possibility of implementing 3D cell culture into drug development pipelines, but also into development of cell-based therapeutics.
Fantastic. And you've just mentioned it, but what kind of impact do you think the 3D cell cultures will have on drug discovery, and how do you see this changing in the future?
If we think about drug development in general, then we need to remember that there is always the so-called valley of death. This is basically the gap between promising initial results in the lab and then lastly, what is translated into the clinics and is then available as a treatment. Many of drugs that are currently being developed don't pass the valley of death. So the development needs to be dropped, and this actually wastes a lot of resources. 3D cell culture is a more physiological system. Therefore, the response to a drug is going to be more likely similar to the response of the patient than what you can achieve in 2D cell culture. So I think by applying modern 3D cell culture pipelines, we can actually reduce the number of compounds that are being lost during development pipelines, which include obviously preclinical phase, then phase 1, phase 2, and so on. In a nutshell, I think 3D cell culture is more predictive to the compared to conventional 2D systems. What's also quite emerging at the moment is development of more complicated 3D cell culture systems where you could cultivate more than just one cell type. We refer to this type of 3D cell culture as organoids. Those are self-organizing systems of cells, and quite recently so-called mini brains have been developed, and those are very good examples of organoids. They resemble a tiny mini brain, and you can also already measure electric activity. Those cells talk to each other and behave sort of like a microbrain. In long term, I think this will certainly change the drug development pipeline and could lead to development of new and groundbreaking therapeutics.
I think that's a really, really interesting area actually, because I myself have seen the development of kind of mini kidneys in a dish and how incredible they are. There's loads of really incredible applications of things like that that could be utilized. One thing I'd love to do is pick your brain, if you will, because I'm just wondering if there are any other really exciting or notable sort of advances in this field that you could tell us about, whether or not that's your own research or just something else that's interesting that you've seen.
What's extremely exciting at the moment is combination of different branches of biomedicine. We have cell culture, we have gene editing, and we have also stem cell technology. And I was really happy to see that all of those cutting edge technologies can actually already now lead to development of new treatment options. Quite recently, a new treatment options have been developed for genetic condition, which results in chronically inflamed skin. And unfortunately, this disease affects small children. And previously, we didn't have any treatment for this syndrome. However, a combination of 3D printing, 3D cell culture, gene editing, and stem cell technology has allowed a few research groups around the globe to develop new option where artificial skin is produced using cell culture, gene editing, and this method has been successfully used in clinical trials. In addition, what I consider quite important are new developments in microfluidics in combination with still 3D cell culture and also organoid technology, resulting in sort of body on a chip. If you think about those systems, you can cultivate organoids from different organs in parallel, connect those to each other by artificial vessels. So this will allow them to talk to each other and study very complex biological systems on a chip, resulting in, again, cost reduction and possibly reducing the number of compounds that we are losing whilst developing new therapies.
I think that's such an exciting area of science, especially the body on a chip or organ on a chip sort of area as well. I think that will be incredible. And then finally, at the end of each podcast, I like to ask the question, which can be a little bit of a tricky one, but where do you see the future of cell culturing heading, or where would you like to see it heading?
I think those are very good questions, and I think those are indeed two very different questions. So where the field is heading at the moment is certainly in the direction of improved and more efficient drug discovery. And this is already being utilized by the whole pharmaceutical industry, where I would like to see the field in, let's say, 10 years is to finally deliver treatment options and new therapies for conditions such as Parkinson's, Alzheimer, where we unfortunately at this stage don't have an answer for, even if we apply most modern cell culture approaches.
Fantastic. Well, I think that's an incredible note to end the podcast. Thank you so much for taking the time to speak with me today. And final question, if people want to get in contact with you, how should they do that?
They could contact me via my official email address, and also they can browse university website where all of my contact details are present.
Perfect. Thanks so much, Darius. It's been brilliant speaking with you.
Thank you very much, and thanks again for inviting me.
Thanks so much for listening along to this incredible episode sponsored by Acrobiosystems. Be sure to subscribe to Opinionated Science on whatever platform you're listening to this episode on, and I look forward to seeing you again for another episode very soon.